Okay, let's dive right in. When you look around, you see all sorts of things living, non-living. Have you ever really stopped to think, what is the fundamental difference? What makes something truly living? What does, say, a plant or an animal have that a rock or a chair just doesn't?
That's such a fundamental question, isn't it? And the answer really boils down to one incredibly important concept: the cell.
The cell, the basic unit of life. That's what we're exploring today, right? Getting into the nitty-gritty of cell, the unit of life.
Yeah.
Using some key biological texts as our guide.
Exactly. Our mission today is to take this foundational, sometimes dense information and make it really clear, really understandable. We want you to have those aha moments about these tiny structures that build everything living.
Sounds great, because understanding the cell is pretty much step one for understanding biology as a whole.
Absolutely. And what's fascinating here is think about how much mystery used to surround life. People saw these complex processes, but didn't know the how or why. It was really the development of a more, let's say, physicochemical approach, sometimes called reductionist biology, that allowed scientists to start peeling back the layers.
Looking at life on a molecular level?
Precisely. Analyzing tissues, figuring out the elements, the organic compounds, what they do inside a cell, how do these molecules drive everything from digestion to memory, even disease. And, we have to acknowledge the intellectual pioneers here, people like G. N. Ramachandran, an absolutely outstanding figure from India.
Ah, yes. His work on protein structure was groundbreaking, wasn't it?
And immensely so. His contributions, like understanding the triple helix of collagen, the Ramachandran plot for protein conformation, they laid critical groundwork for how we even approach molecular biology today. It showed how complex functions arise from these, understandable molecular interactions.
So that journey into the molecular world really starts with understanding the cell itself. Where do we begin with that? The basic definition.
Let's start right there. Section 1.
Yeah.
The foundational concept. Okay, the definition. Simplest terms. The cell is the basic unit of life.
Basic unit.
Meaning it's the smallest thing that can be considered truly alive. It has two key capabilities you absolutely need to grasp. First, independent existence. It can survive on its own under the right conditions, of course.
Okay, independent existence.
And time. Second, it can perform all the essential functions of life. Think metabolism, growth, reproduction, responding to the environment, all the core processes.
So whether it's a single-celled organism like bacteria.
Exactly, a unicellular organism.
Or a complex multicellular one like us, it's all built from these units that can potentially live and function independently.
Precisely. And here's a really interesting point to consider. Anything less than the complete structure of a cell just doesn't cut it for independent living.
Like a virus. They need a host cell, right?
Perfect example. A virus has genetic material, a protein code, but it lacks the machinery to replicate or carry out metabolism on its own. It must hijack a host cell. It doesn't meet that independent existence and function criteria. That really reinforces why the cell is so fundamental.
Okay, that makes sense. But these things are tiny, invisible. How on earth did we even find out they existed?
Ah, that's a journey into the history of science and technology. It all hinges on the invention of the microscope.
Right, you can't see what you don't have the tools to look for.
Exactly. For centuries, this whole microscopic world was completely hidden. Then, in the late 1600s, you have Anthony von Leeuwenhoek, a Dutch draper, not even a formally trained scientist initially, but obsessed with lens grinding.
He made his own microscopes.
He did. And they were remarkably powerful for the time single-lens microscopes. He was the first person to actually see and describe living cells. He saw bacteria, protozoa, call them animalcules. Imagine seeing that for the first time.
Mind-blowing, really. Opening up a whole new reality.
Absolutely revolutionary. Then, much later, in 1831, Robert Brown, a botanist, noticed a consistent dense spot inside plant cells.
And that was the nucleus.
That was the nucleus, another key piece of the puzzle. And of course, the ongoing improvements in microscopes, eventually leading to the electron microscope in the 20th century, just kept revealing more and more intricate detail inside the cell.
So Leeuwenhoek sees cells, Brown sees the nucleus. How did these observations coalesce into the actual cell theory? It wasn't immediate, was it?
No, not at all. It took time and different minds working on it. The formal theory really started taking shape in the 1830s. You had Matthias Schleiden, a German botanist, in 1838.
What did he observe?
He looked at a huge number of plants and came to a general conclusion. All plants seem to be composed of different types of cells, and these cells form the plant tissues, a big generalization for the plant world.
Okay, so that covers plants. What about animals?
Right. Then, almost immediately after, in 1839, another German scientist, Theodor Schwann, a zoologist this time, was doing similar work on animal cells. He noticed that animal cells consistently had this thin outer layer, what we now call the plasma membrane.
And he compared them to plant cells.
He did. Drawing on Schleiden's work and his own observations, Schwann noted that plant cells had a rigid cell wall outside the membrane, which animal cells lacked. That was a key difference.
So he saw similarities and differences.
Yes. And based on all this, Schwann proposed a really bold hypothesis that the bodies of both animals and plants are fundamentally composed of cells and the products of cells. That was the crucial unifying idea.
So Schleiden and Schwann basically put their findings together.
They did. They collaborated, discussed, and formulated the initial version of the cell theory, a monumental step. It stated that organisms are made of cells.
But there was a gap, wasn't there? Something missing.
There was a big one. Their theory was great at describing what organisms were made of, but it didn't explain where new cells came from. Did they just spontaneously appear? That was the missing piece.
Ah, okay. So how is that resolved?
That critical refinement came a bit later, in 1855, from Rudolf Virchow, another German physician and scientist. He made a powerful and concise statement, famously in Latin, omnis cellula cellula, meaning all cells arise from pre-existing cells. Simple, elegant, but profoundly important. It dismissed the idea of spontaneous generation for cells.
So new cells only come from existing cells dividing.
Exactly. Cell division. That was the key. Virchow's modification completed the cell theory, giving it the final shape we accept today.
And the core tenets are?
2 main points to always remember. 1, all living organisms are composed of cells and the products of cells. And 2, all cells arise from pre-existing cells.
That really establishes the continuity of life, doesn't it? Everything comes from something before it, all the way back.
Precisely. It underlines the fundamental unity of all life, despite the incredible diversity we see. It's the bedrock of modern biology.
Okay, so we have the theory. Now let's get a bit more concrete. Let's look inside the cell, get an overview, maybe start with some common examples, like the onion peel cells you mentioned.
Good idea. Thinking about what you might actually see under a basic microscope helps. If you look at onion peel cells, they're plant cells. You'll clearly see that outer boundary, the rigid cell wall, and just inside that, there's the cell membrane.
Okay, plant cell, wall, then membrane. What about an animal cell, like from a human cheek scraping?
Right. If you look at cheek cells, they're animal cells. What you see is the outer boundary is the plasma membrane. There's no cell wall there. That's a fundamental difference right away.
And inside both types, we find that structure Brown discovered, the nucleus?
Generally, yes. Inside typical plant animal cells, you'll see a prominent, dense, usually spherical structure, the nucleus. And this is crucial because the nucleus contains the cell's genetic blueprints, the chromosomes, which are made of DNA.
And the presence or absence of this nucleus is a major dividing line, isn't it?
Absolutely fundamental. This leads us to the 2 major categories of cells. Cells that do have this well-defined membrane-bound nucleus are called eukaryotic cells. Think eu, meaning true, and karyon, meaning nucleus. True nucleus.
Eukaryotic, like plants, animals, fungi, protists.
Exactly. All those are eukaryotes. Now, cells that lack a membrane-bound nucleus where the genetic material is sort of floating more freely in the cell interior are called prokaryotic cells. Pro meaning before a true nucleus.
Prokaryotic, like bacteria and archaea.
Precisely. Bacteria, blue-green algae, cyanobacteria, those are prokaryotes. This distinction, membrane-bound nucleus or not, is probably the single most important division in cell biology.
Okay. Nucleus or no nucleus. What about the rest of the stuff inside, the jelly?
Ah, the cytoplasm. Yes, in both prokaryotes and eukaryotes, the cell is filled with this semi-fluid matrix called the cytoplasm. You could think of it as the cell's internal environment, the main stage where all the action happens.
The action being?
All the biochemical reactions that keep the cell alive. Metabolism, building molecules, breaking them down.
Yeah.
It's the bustling workshop of the cell, the main arena of cellular activities, as the texts often say.
So if cytoplasm is the general arena, do eukaryotes have more specialized sections within that arena compared to prokaryotes?
That's a perfect way to put it. Eukaryotic cells are all about compartmentalization.
Yeah.
Besides the nucleus, their cytoplasm is filled with various other distinct membrane-bound structures called organelles.
Like little organs within the cell.
Exactly, each with a specific job. We're talking about the endoplasmic reticulum, ER, the Golgi complex, lysosomes, mitochondria, vacuoles, microbodies. All these compartments allow for different chemical environments and processes to occur simultaneously without interfering with each other. It's highly organized.
And prokaryotes just don't have those?
Largely, no. Prokaryotic cells lack these complex membrane-bound organelles. Their internal organization is much simpler. That's a key difference.
Is there anything found in both besides the cytoplasm and plasma membrane?
Yes, one very important structure, ribosomes. Ribosomes are responsible for making proteins, following the instructions from the DNA. Critically, ribosomes are not bound by a membrane.
Ah, so because they're not membrane bound, they can be in both types of cells.
Exactly. You find ribosomes in prokaryotes floating in the cytoplasm, and in eukaryotes you find them in the cytoplasm too, but also attached to the ER, and even inside some other organelles like mitochondria and chloroplasts.
Interesting. Any other non-membrane bound things?
In eukaryotes, specifically animal cells, there's also the centrosome, which helps organize microtubules during cell division. It's usually absent in plant cells. But ribosomes are the truly universal non-membrane-bound organelle.
Okay, so there's this fundamental split, prokaryotic simplicity versus eukaryotic complexity and compartmentalization. But even within those groups, cells vary wildly, don't they? In size, shape, what they do.
Oh, absolutely. The diversity is staggering. Let's talk size first. You have mycoplasmas, which are basically tiny bacteria, perhaps the smallest things you could call cells, only about 0.3 micrometers long. Barely visible.
So really tiny. And on the other end,
Bacteria themselves are usually around 3 to 5 micrometers. But then you jump to the other extreme. The largest isolated single cell is the egg of an ostrich, something you can actually see and hold.
wow. And what about cells within one organism, like humans?
Huge variation there too. Human red blood cells are tiny discs, about 7 micrometers across. But then you have nerve cells, neurons, which can be incredibly long, some stretch from your spinal cord all the way down to your foot, over 1 meter long, potentially.
A single cell that long. That's amazing. And shape varies just as much.
Definitely. You see all sorts. Red blood cells are disc-like, biconcave. Some cells are polygonal, like paving stones. Epithelial cells lining your gut might be columnar or cuboid. Plant cells involved in water transport can be long and thread-like. White blood cells can be irregular, changing shapes as they move.
So it's not random. The shape relates to the job.
Absolutely. That's the key principle. The shape of the cell generally varies with the function it performs. The long nerve cell transmits signals over distance. The flat skin cell provides covering. The irregular white blood cell engulfs invaders. Form follows function, right down to the cellular level.
Right, that makes sense. So let's dive deeper into the first group, the prokaryotes. Simpler, yes, but incredibly important and diverse.
Absolutely. This group includes organisms you've definitely heard of: bacteria, blue-green algae or cyanobacteria, mycoplasma, and things called PPLO pleuropneumonia, like organisms which are essentially tiny wall-less bacteria.
Key features.
Generally, they are much smaller than eukaryotic cells, and they tend to multiply much more rapidly. Think about how quickly a bacterial infection can take hold that's partly due to their rapid division rate.
And they come in different shapes, too.
Yes, bacteria have characteristic shapes that are used for classification. You have
Right.
bacillus, rod-shaped; coccus, spherical; vibrio, comma-shaped; spirillum, spiral-shaped. But despite these different shapes and lifestyles...
They share a fundamental organization.
Exactly. If we connect this to the bigger picture, all prokaryotes, despite their outward variety, have a remarkably similar underlying structure. They all have a cell membrane. Most, except mycoplasma, have a cell wall outside that. Inside is the cytoplasm.
And the genetic material.
No well-defined nucleus, remember. The genetic material, usually a single circular DNA molecule, is located in a region called the nucleoid. It's not enclosed by a membrane.
Naked DNA, essentially.
Essentially. And here's something really significant, especially for things like antibiotic resistance. Many bacteria also have small, extra circles of DNA floating in the cytoplasm, outside the main chromosome. These are called plasmids.
Plasmids, right. Those are crucial. They can carry genes for things like resistance.
Precisely. Plasmids can carry genes that give the bacterium unique traits, like the ability to resist certain antibiotics. This is a huge issue in medicine, obviously. They can even be transferred between bacteria, spreading resistance.
And they're used in genetic engineering too, right, as tools?
Absolutely. Because they're small and easy to manipulate, scientists use plasmids as vectors to carry foreign DNA into bacteria, a cornerstone of biotechnology. So remember, prokaryotes, no true nucleus, usually circular DNA in a nucleoid, often plasmids, and generally no complex membrane-bound organelles.
Okay, Let's talk about the outer layers, the cell envelope. You said most have a cell wall. What else is there?
Right, the cell envelope. In most prokaryotes, especially bacteria, it's a complex, multi-layered structure, typically 3 layers, tightly bound together. 1, outermost, glycocalyx. 2, middle, cell wall. 3, innermost, plasma membrane. They function together as a single protective unit.
Glycocalyx, that sounds like a sugary coat.
It often is. It varies, though. In some bacteria, it's a loose, slimy layer, just called the slime layer, easy to wash off. In others, it's thick, tough, and well-organized, forming a capsule.
Does the capsule do anything special?
Oh, yes. Capsules often help bacteria resist being engulfed by immune cells, making them more virulent. They can also help bacteria stick to surfaces. The nature of this outer layer, along with the cell wall, is actually key to how bacteria are classified using Gram staining.
Ah, gram-positive and gram-negative.
Exactly, developed by Hans Christian Gram. Bacteria that retain the crystal violet stain appear purple and are gram-positive. Those that don't retain it and are counterstained pink are gram-negative. This difference reflects fundamental differences in their cell envelope structure, particularly the thickness and composition of the cell wall and the presence or absence of an outer membrane in gram-negatives.
Okay, so the glycocalyx varies. What's the cell wall's main job?
The cell wall just inside the glycocalyx is critical. It provides shape, structural support like scaffolding, and very importantly, it prevents the cell from bursting when water rushes in, osmotic lysis, or collapsing if water leaves. It maintains the cell's integrity.
And the innermost layer, the plasma membrane.
The plasma membrane is the gatekeeper. Just like in eukaryotes, it's selectively permeable, controlling what gets in and out. It's where many metabolic processes occur, and it's structurally quite similar to the eukaryotic plasma membrane.
Now, you mentioned a unique prokaryotic structure earlier, the mesosome. What's that about?
Ah, yes, the mesosome. This is fascinating and unique to prokaryotes. It's essentially an infolding of the plasma membrane into the cytoplasm.
So the membrane folds inward.
Right, these folds can look like little sacs, vesicles, tubes, tubules, or layered sheets, lamellae.
And what do these infoldings do? It seems like a simple structure to have so many functions attributed to it.
That's the intriguing part. They seem to be involved in a surprising number of crucial processes, perhaps compensating for the lack of other organelles. Functions include helping with cell wall synthesis, playing a role in DNA replication and separating the DNA into daughter cells during division, containing enzymes for respiration, sort of like primitive mitochondria, involvement in secretion, and generally increasing the surface area of the membrane for enzymatic activity. In some photosynthetic prokaryotes, like cyanobacteria, you find other types of internal membrane extensions called gluconophores, which hold the photosynthetic pigments.
Okay, so they have these internal membrane folds. What about movement? Do prokaryotes move?
Many do. If a bacterium is motile, it usually possesses flagella, singular flagellum. These are long, thin, whip-like appendages extending from the cell wall.
Like little propellers.
Pretty much. The number and arrangement of flagella vary between bacterial species. Each flagellum is a complex structure itself, composed of 3 parts. The long filament extending outwards, a hook connecting it, and a basal body embedded in the cell envelope that acts as a rotary motor.
Are there other things sticking out from the surface? Maybe not for moving, but for sticking to things.
Yes, good question. Besides flagella, bacteria can have 2 other types of surface appendages.
Yeah.
Pili, singular, pilis, and fimbriae, singular fimbria. Critically, these are not involved in motility.
Not for movement, so what are they for?
Piles are typically longer, tubular structures made of protein, and there are usually only a few per cell. Some specialized pili are involved in bacterial conjugation transferring genetic material. Fimbriae are shorter, thinner, bristle-like fibers, often very numerous.
And their main job?
Attachment. Fimbriae are crucial for helping bacteria adhere to surfaces like rocks in a stream, or, importantly for disease, to the tissues of a host organism. Think of them as tiny grappling hooks.
Okay, attachment structures. Now let's get back inside. We mentioned ribosomes, the protein factories. Where are they in prokaryotes?
Right, ribosomes. In prokaryotes, they are often found associated with the plasma membrane, as well as free in the cytoplasm. They are small, about 15 by 20 nanometers.
And their structure, size?
They're made of ribosomal RNA, rRNA, and proteins. Prokaryotic ribosomes are designated 70S. This 70S ribosome is actually made of 2 subunits, a larger 50S subunit and a smaller 30S subunit.
50 plus 30 doesn't equal 70.
Exactly. The S stands for Svedberg unit, a measure of sedimentation rate in a centrifuge, related to size and shape, not simple mass addition. So 50S plus 30S combine to make a 70S functional ribosome. Their job, of course, is protein synthesis, translating the genetic code from messenger RNA, mRNA, into proteins.
And sometimes multiple ribosomes work on the same message.
Yes. Often several ribosomes attached to a single mRNA molecule, forming a chain called a polyribosome or polysome. This allows the cell to produce many copies of the same protein very quickly and efficiently from one mRNA template.
Efficient protein production. Now, you also mentioned prokaryotes store materials differently. How do they do that without compartments? This raises an important question about storage in simple cells.
Good point. They use structures called inclusion bodies. Since they lack membrane-bound organelles for storage, reserve materials accumulate in the cytoplasm as these non-membrane-bound granules or aggregates.
Examples.
Things like phosphate granules, cyanofissin granules in cyanobacteria, possibly nitrogen storage, and glycogen granules for carbohydrate storage. They just lie freely in the cytoplasm.
Any others?
Some aquatic prokaryotes, like certain photosynthetic bacteria, blue-green, purple-green bacteria, have gas vacuoles. These aren't true vacuoles with membranes, but rather clusters of gas-filled vesicles that help regulate buoyancy, allowing the bacteria to float at the optimal depth for sunlight.
Okay, so prokaryotes are simple in structure but remarkably efficient and diverse. That sets the stage nicely for the complexity of the eukaryotes. All right, let's shift gears to eukaryotic cells. This is where things get significantly more intricate, the world of compartmentalization.
Indeed. Eukaryotes include a vast range of organisms, protists, fungi, plants, and animals. Their hallmark, as we've touched on, is this extensive compartmentalization. The cytoplasm is divided up by numerous membrane-bound organelles.
Plus, they have that organized nucleus with a proper nuclear envelope.
Exactly, and typically more complex structures for movement, locomotory structures, like the eukaryotic cilia and flagella we'll discuss, and an internal protein framework, the cytoskeleton. Their genetic material, DNA, is also highly organized into multiple linear chromosomes, usually complex with proteins. This compartmentalization allows for much greater specialization and efficiency.
Now, what's fascinating here is that even within this complex eukaryotic world, there are key differences, especially between plant and animal cells. What are the main ones we need to keep straight?
Yes, the classic plant versus animal cell comparison. Crucial differences to remember. Plant cells have a rigid cell wall outside the plasma membrane. Plant cells have plastids, like chloroplasts for photosynthesis. Plant cells have typically a large central vacuole for storage and turgor pressure. Animal cells, on the other hand, lack a cell wall. Animal cells, on the other hand, lack plastids. Animal cells, on the other hand, lack a large central vacuole, may have small temporary ones. Animal cells, on the other hand, do possess centrioles involved in cell division, which are absent in almost all plant cells.
Okay, those are the big headline differences. Now let's zoom in on the individual components, starting with the boundary itself.
The plasma membrane.
Or cell membrane. It's the outer boundary of animal cells and lies just inside the cell wall in plant cells. Its detailed structure really only became clear with the electron microscope in the 1950s.
But early chemical studies gave clues, right? Especially on red blood cells. They did. Those studies showed it was primarily composed of lipids and proteins. The main lipids are phospholipids.
And they arrange themselves in that specific way. The bilayer.
Exactly, the lipid bilayer. The phospholipids have a hydrophilic, water-loving polar head and a hydrophobic, water-fearing nonpolar tail. They arrange themselves spontaneously in water with the heads facing outwards towards the watery exterior and interior of the cell, and the tails tucked inwards, away from the water.
Creating a barrier that's oily on the inside.
Precisely, a barrier to water-soluble substances. Eukaryotic membranes also contain cholesterol interspersed among the phospholipids, which affects the membrane's fluidity. And later studies showed proteins are major components too. The ratio varies. Human red blood cells are about 52% protein, 40% lipid by weight, but it depends on the cell type and its function.
Are the proteins just stuck on the surface?
Not necessarily. Based on how easily they can be removed, proteins are classified as peripheral proteins, loosely associated with the surface; integral proteins, more deeply embedded, sometimes partially, sometimes fully spanning the entire bilayer, transmembrane proteins. These often act as channels or transporters.
Okay, so lipids, proteins, cholesterol, how does it all fit together? This brings us to that famous model, right? The fluid mosaic model.
Yes, the fluid mosaic model, proposed by Singer and Nicholson in 1972, it's the accepted model. It describes the membrane not as a static structure, but as dynamic and fluid.
Fluid meaning?
Meaning the components can move. The model proposes a quasi-fluid nature for the lipids, allowing the lipids themselves, and importantly many of the proteins, to move laterally within the plane of the membrane. Think of it like proteins floating or drifting in a sea of lipids.
A mosaic that can shift and rearrange.
Exactly. This fluidity is crucial. It's not just an abstract property. It's essential for many cell functions.
Why is fluidity so critical? What does it allow the cell to do?
Think about it. A rigid wall couldn't grow, divide, or let things in easily. Fluidity is essential for cell growth, forming junctions between cells, secretion, releasing substances, endocytosis, taking substances in, cell division, and just generally allowing proteins to interact and function properly within the membrane.
So it's dynamic, and one of its most important jobs is controlling passage, being selectively permeable. How does that work?
Right. Selective permeability is key. The membrane doesn't let everything through equally. Transport across the membrane happens in several ways. First, passive transport. Doesn't require the cell to expend energy. Molecules move down their concentration gradient, high to low. Simple diffusion. Small nonpolar molecules like oxygen, CO2, can slip directly through the lipid bilayer. Osmosis. The specific diffusion of water across the membrane, also down its water potential gradient.
But what about things that can't just slip through the oily layer, like ions or sugars?
Good point. Polar molecules and ions can't easily cross the hydrophobic core. They need help. This leads to facilitated diffusion. Still passive, no cell energy used, still down the gradient, but it requires a carrier protein or channel protein embedded in the membrane to help the substance across.
Like a specific doorway.
Exactly. And then there's the case where the cell needs to move something against its concentration gradient from low concentration to high.
Pushing it uphill, essentially. That must require energy.
It absolutely does. This is active transport. It requires cellular energy, usually from ATP, to pump substances against their gradient. The classic example is the sodium-potassium Na plus K plus pump, vital for nerve cells and others. It pumps sodium ions out and potassium ions in, both against their gradients.
Okay, that covers the plasma membrane. Now, in plants and fungi, there's that extra layer outside the cell wall. What's its role?
The plant cell wall is a rigid, non-living outer layer. It provides, shaped to the plant cell, protection from mechanical stress and pathogens, prevents excessive water uptake that could burst the cell, plays a role in cell-to-cell interaction, acts as a barrier to large, potentially harmful macromolecules.
Is it always made of the same stuff?
Mostly cellulose in higher plants, often combined with hemicellulose, pectins, and proteins. Algae have variations, cellulose, galactins, mannans, even calcium carbonate.
And it can grow.
Yes, the initial primary wall laid down in a young growing cell is relatively thin and extensible. As the cell matures and stops growing, a thicker, more rigid secondary wall is often deposited on the inner side of the primary wall.
Now, what's fascinating here is how plant cells, despite these rigid walls, manage to stick together and communicate. How does that work?
Great question. Plant cells in a tissue are cemented together by the middle lamella. This is a layer rich in calcium pectate that lies between the primary walls of adjacent cells, effectively gluing them together.
Like mortar between bricks.
Good analogy. And for communication through the walls, they have plasmodesmata. These are tiny channels that directly connect the cytoplasm of adjacent plant cells, passing through the cell walls and middle lamella.
So direct cytoplasmic connections.
Yes, allowing water, small cellulose, and even larger molecules like signaling proteins to pass directly from cell to cell, essential for transport and coordination in plants.
Okay, moving inward again. You mentioned earlier that many organelles and eukaryotes work together. This brings us to the endomembrane system. What's included in that?
Right, the endomembrane system. This is a group of membranes and organelles in eukaryotic cells that work together in the synthesis, processing, packaging, and transport of proteins and lipids. Think of it as an integrated network. The key players are the endoplasmic reticulum, ER, the Golgi apparatus or Golgi complex, lysosomes, vacuoles. Their membranes and functions are interconnected either through direct physical contact or by the transfer of membrane segments as vesicles.
And just to be clear, what's not part of this system?
Good clarification. Mitochondria, chloroplasts, and peroxisomes are generally not considered part of the endomembrane system. Although they are membrane-bound, their functions, energy production, photosynthesis, specific metabolic reactions, are largely independent of this ER-Golgi pathway. They don't directly participate in that flow of materials.
Okay, system components. Let's start with the ER endoplasmic reticulum. It sounds like a network.
It is a vast network of membranes forming sacs, cisternae, and tubules that extends throughout the cytoplasm of eukaryotic cells. It was revealed in detail by electron microscopy. The ER membranes essentially divide the cell's interior into two compartments: the luminal space, inside the ER sacs and tubules, and the extraluminal space, the cytoplasm outside.
And there are two types based on appearance, rough and smooth.
Exactly. Rough endoplasmic reticulum, RER, appears rough because its outer surface is studded with ribosomes. It's heavily involved in synthesizing proteins that are destined for secretion, insertion into membranes, or delivery to other organelles like the Golgi or lysosomes. It's often continuous with the outer membrane of the nuclear envelope. Smooth endoplasmic reticulum, SER. Lacks ribosomes, so it looks smooth. It's involved in different functions, primarily lipid synthesis, including steroids like hormones, detoxification of drugs and poisons, especially in liver cells, and calcium storage.
So proteins from the RER and lipids from the SER then move on to the Golgi apparatus.
That's the typical flow, the Golgi apparatus, or Golgi complex body, named after Camillo Golgi, who first described it in 1898. It looks like a stack of flattened, membrane-bound sacs called cisternae. Think of a stack of pancakes, but curved.
A stack of cisternae, and it has distinct sides.
Yes, this is crucial for its function. The Golgi stack has polarity. The cis face, or forming face, usually convex and oriented towards the ER. It receives vesicles containing newly synthesized proteins and lipids from the ER. The trans face, or maturing face, usually concave and oriented away from the ER. It's where processed molecules are packaged into new vesicles to be shipped off.
So if we connect this to the bigger picture, the Golgi is like the cell's post office or processing and packaging center.
Exactly. Materials arrive from the ER at the cis face, move through the Golgi stack from cisterna to cisterna, and undergo modification and sorting along the way.
What kind of modifications?
Proteins might be further folded, have carbohydrate chains added, forming glycoproteins, or be cleaved. Lipids can also be modified, perhaps by adding carbohydrates, forming glycolipids. The Golgi ensures molecules are properly processed and tagged for their final destination, whether that's secretion outside the cell, delivery to the plasma membrane, or transport to another organelle like the lysosome. Then they bud off in vesicles from the transphase.
Okay, processing and packaging. That leads us to lysosomes. They come from the Golgi, right? And they're the recycling centers.
Correct on both counts. Lysosomes are membrane-bound sacs containing hydrolytic enzymes. They bud off from the transphase of the Golgi.
Hydrolytic enzymes meaning they break things down using water?
Precisely. Lysosomes are filled with enzymes like proteases, break down proteins, nucleuses, break down nucleic acids, lipases, lipids, and carbohydrates. These enzymes work best at an acidic pH, which is maintained inside the lysosome.
And their job?
Digestion and recycling. They break down ingested materials, like food particles in a food vacuole, cellular debris, and old, worn-out organelles. They're essential for cellular housekeeping.
All right. The last part of the endomembrane system you listed was vacuole. Big in plants, you said.
Very big in mature plant cells. Vacuoles are membrane-bound sacs, but the membrane surrounding a vacuole has a specific name, the tonoplast.
Tonoplast. And what's inside?
Varies depending on the cell and type of vacuole. Can contain water, ions, pigments, waste products, stored nutrients, defensive compounds. In plant cells, the large central vacuole can occupy up to 90% of the cell volume. It stores water, maintains turgor pressure, keeping the cell firm, stores reserves, and sequesters waste.
What's fascinating here is how the tonoplast works. You mentioned it transports things against the gradient into the vacuole.
Yes, the tonoplast actively transports certain solutes, like ions, from the cytoplasm into the vacuole, even if the concentration inside is already higher. This requires energy, but allows the plant cell to accumulate high concentrations of substances and helps maintain turgor.
Do animal cells have vacuole?
They can, but they are usually much smaller, more numerous, and temporary than the large central vacuole of plants. Their functions are more varied: temporary storage, transport, waste disposal. Some protists have specialized vacuoles, like the contractile vacuole in amoeba for pumping out excess water, or food vacuoles formed during phagocytosis.
Okay, that wraps up the endomembrane system. Let's move to the energy specialists. First, mitochondria, the powerhouses.
Right. Mitochondria,
Yeah.
singular, mitochondrion, found in nearly all eukaryotic cells. They are the primary sites of aerobic cellular respiration.
Meaning they use oxygen to generate.
ATP, adenosine triphosphate, the main energy currency of the cell. That's why they're called the powerhouses. Their number varies greatly. Cells with high energy demands, like muscle or liver cells, have lots of them. They're typically sausage-shaped or cylindrical.
Structure-wise, they have two membranes.
Yes, a crucial feature. Mitochondria are enclosed by two membranes, a smooth outer membrane, a highly folded inner membrane. The space between these is the intermembrane space. The intermembrane folds inwards to form shelf-like structures called cristae.
And the cristae are important because
They dramatically increase the surface area of the inner membrane. This is critical because many of the enzymes and protein complexes involved in ATP synthesis, the electron transport chain in ATP synthesis, are embedded within this inner membrane. More surface area involves more space for energy production machinery.
makes sense. And the innermost space is called
The mitochondrial matrix. This is the space enclosed by the inner membrane. It contains mitochondrial DNA, ribosomes, and enzymes for the Krebs cycle, another key part of respiration.
Wait, mitochondria have their own DNA and ribosomes?
They do. The matrix contains a small, usually circular DNA molecule and 70S ribosomes, like prokaryotic ones. This is strong evidence for the endosymbiotic theory, the idea that mitochondria originated as free-living bacteria that were engulfed by an ancestral eukaryotic cell billions of years ago.
So they're like semi-autonomous guests within the cell.
In a way, yes. They can synthesize some of their own proteins and even replicate by fission, similar to bacteria. Fascinating evolutionary story.
Okay, from mitochondria to plastids, these are mainly in plants and algae, right? The color and kitchen of the cell?
Good description. Plastids are found in plant cells and euglenoids. They often contain pigments, giving color, and are involved in synthesis and storage. Based on their pigments and function, they are classified into three main types: chloroplasts, chromoplasts, and leucoplasts.
Let's break those down. Chloroplasts are the most famous for photosynthesis.
Exactly. Chloroplasts contain chlorophyll, the green pigment, and carotenoid pigments, yellow-orange. They trap light energy and convert it into chemical energy, sugars, via photosynthesis. They are most abundant in the green parts of plants, especially the mesophyll cells of leaves.
Structure, also double membraned.
Yes, like mitochondria, chloroplasts have a double membrane, outer and inner. The fluid-filled space inside the inner membrane is called the stroma.
And inside the stroma?
Within the stroma is a complex system of interconnected membranous sacs called thylakoids. Thylakoids are often stacked like coins into structures called grana, singular, granum. The thylakoid membranes contain the chlorophyll and other pigments and are the sites of the light-dependent reactions of photosynthesis.
And the stroma itself.
The stroma contains enzymes for the light-independent reactions, Calvin cycle, where CO2 is converted into sugars. And just like mitochondria, the stroma also contains the chloroplast's own circular DNA and 70S ribosomes, again pointing to an endosymbiotic origin.
So chloroplasts handle the green. What about other colors?
That's where chromoplasts come in. They synthesize and store carotenoid pigments, like carotene and xanthophylls, which give fruits, flowers, and autumn leaves their yellow, orange, and red hues. They help attract pollinators and seed dispersers.
And the third type, colorless.
Leucoplasts. These are non-pigmented plastids, mainly involved in storage. They come in different subtypes. Amyloplasts, store starch, Eugene, potatoes' roots. Haleoplasts, store oils and fats. Aleuroplasts, store proteins.
Okay, a whole family of plastids with different jobs. Now let's revisit ribosomes. We know they make protein and are in both prokaryotes and eukaryotes, discovered by Palade.
Yes, George Palade identified them with the electron microscope in the 1950s. They are granular structures made of ribosomal RNA, rRNA, and proteins. Key feature: no surrounding membrane.
And the size difference, 70S versus 80S. Can you clarify the S again?
Right. Eukaryotic ribosomes in the cytoplasm and on the RER are larger, designated 80S. They consist of a 60S large subunit and a 40S small subunit. Prokaryotic ribosomes, and those in mitochondria and chloroplasts, are smaller, 70S. They consist of a 50S large subunit and a 30S small subunit. S stands for Svedberg unit, a measure of sedimentation rate during centrifugation. It reflects mass and shape, so the subunit's S values don't simply add up. This size difference is clinically important. Many antibiotics target bacterial 70S ribosomes, inhibiting their protein synthesis without harming our 80S ribosomes.
Got it. Now, eukaryotic cells also have an internal support structure, don't they? The cytoskeleton.
Yes, the cytoskeleton. It's an intricate network of protein filaments extending throughout the cytoplasm. Think of it as the cell's internal scaffolding and railway system.
Made of what?
Three main types of protein filaments. Microtubules, hollow tubes made of tubulin protein, the thickest component. Microfilaments, solid rods made primarily of actin protein, the thinnest. Intermediate filaments, fibrous proteins coiled into cables, intermediate in diameter.
And its functions.
It provides mechanical support and maintains cell shape. It's involved in cell motility, movement, and the movement of organelles within the cell. It also plays a role in anchoring organelles and in cell division. It's very dynamic, constantly assembling and disassembling.
Speaking of movement, let's look at cilia and flagella in eukaryotes, hair-like extensions.
Exactly. Cilia, singular dot, something, are projections from the cell surface involved in movement. Cilia are usually short and numerous. They beat like oars, either propelling the cell, like paramecium, or moving fluid over the cell surface, like in our respiratory tract. Flagella are typically much longer and fewer, often just one or two per cell. They have a more whip-like or undulating motion to propel the cell, like sperm.
And importantly, these eukaryotic versions are structurally different from prokaryotic flagella.
Very different internally, yes. A key distinction.
So what is the internal structure, that famous 9+2 array?
Yes, the classic 9+2 arrangement. If you cut a eukaryotic cilium or flagellum cross-sectionally and look with an electron microscope, you see it's covered by an extension of the plasma membrane. Inside is the core, called the axonome. The axonome consists of microtubules arranged in a precise pattern, 9 doublets of microtubules arranged in a ring around the periphery, and 2 single microtubules in the center. That's the 9+2.
9 pairs outside, 2 singles inside.
Correct. And these microtubules are interconnected by various proteins, linkers between the doublets, radial spokes connecting the outer doublets to the central pair, and motor proteins like dynein that cause the bending motion. This whole structure arises from a basal body at the base, which has a structure similar to a centriole.
Which brings us nicely to the centrosome and centrioles, found mainly in animal cells.
Yes, the centrosome is the main microtubule organizing center in animal cells. It typically consists of 2 centrioles oriented perpendicular to each other, surrounded by amorphous pericentriolar material.
And the centrioles themselves have a unique structure, the cartwheel.
They do. Each centriole is a cylindrical structure made of 9 triplets of microtubules arranged in a ring. No central microtubules here, a 9+0 arrangement essentially for the main structure. These triplets are linked, and radial spokes connect them to a central proteinaceous hub, giving it that cartwheel appearance in cross-section.
What are their main functions?
2 key roles. 1, they form the basal bodies from which cilia and flagella grow. 2, during animal cell division, the centrosome replicates, and the 2 centrosomes move to opposite poles of the cell, organizing the microtubules of the spindle apparatus, which is essential for separating chromosomes.
Okay, almost there. The big one. The nucleus. The control center. Discovered by Brown, chromatin named by Fleming. Let's get into its structure.
Right, the nucleus. It contains the cell's main genetic material and controls the cell's growth and reproduction. In a non-dividing interphase cell, the nucleus contains chromatin, a complex of DNA and proteins, mostly histones, that forms the chromosomes. In interphase, it's dispersed, like tangled threads. Nucleolus, or nucleoli, dense structures involved in ribosome synthesis, nucleoplasm, the fluid matrix filling the nucleus, all enclosed by the nuclear envelope.
The envelope is double, right? 2 membranes.
Yes, a double membrane consisting of 2 parallel membranes separated by the perinuclear space. The outer membrane is often continuous with the RER and may have ribosome.
And it has pores.
Crucially, yes. The envelope is perforated by numerous nuclear pores. These aren't just simple holes. They are complex protein structures that regulate the transport of molecules, like RNA out, proteins in, between the nucleus and the cytoplasm. Very selective gateways.
Usually 1 nucleus per cell, but sometimes more, or even none.
Typically 1, yes. But some cells are multinucleated, like skeletal muscle cells. And as we mentioned, some mature cells, like mammalian red blood cells and plant sieve tube cells, lose their nucleus entirely. They function, but have limited lifespans and capabilities.
Inside the nucleoplasm, you mentioned the nucleolus. What's its specific job?
The nucleolus is the site of ribosomal RNA synthesis, and the assembly of ribosomal subunits. It's not membrane-bound. Cells actively making lots of protein usually have prominent nucleoli.
And the chromatin, the DNA protein complex, it condenses during cell division.
Exactly. During interphase, chromatin is extended and diffuse, allowing access for gene transcription. But when the cell prepares to divide, the chromatin undergoes dramatic condensation to form distinct, visible chromosomes. This makes the genetic material manageable for segregation.
And chromatin is DNA wound around proteins.
Primarily DNA wound around basic proteins called histones, which helps package the incredibly long DNA molecule into a compact form. There are also non-histone proteins involved in regulation. Think of that 2 meters of DNA in each human cell needing to fit inside a tiny nucleus.
Wow. So a chromosome structure typically involves.
After DNA replication before division, a chromosome usually consists of 2 identical sister chromatids joined at a constricted region called the centromere. On the centromere are protein structures called kinetochores, where spindle fibers attach during division.
And chromosome shape depends on where that centromere is.
Yes. We classify them based on centromere position. Metacentric, a centromere in the middle, equal arms. Submetacentric, off-center, one short, one long arm. Acrocentric, your one end, one very short, one long arm. Telecentric, at the very end, essentially one arm. Some chromosomes also have secondary constrictions and tiny fragments called satellites beyond them.
Okay, one last tiny structure, microbodies.
Yes, microbodies. These are small, membrane-bound vesicles containing various enzymes. Examples include peroxisomes, involved in breaking down fatty acids and detoxifying harmful substances like hydrogen peroxide, and kylioxysomes, in plants, help convert stored fats to carbohydrates during seed germination. They compartmentalize specific metabolic reactions. Hashtag tag atrazine.
Wow. Okay, that was a deep dive indeed. So let's try to bring it all together. We started asking what makes something alive, and the answer really is the incredible complexity and organization within the cell.
It really is. We saw how the cell theory developed, establishing that all life is cellular and that cells come from pre-existing cells, a unifying principle.
And we contrasted the fundamental simplicity of prokaryotes with the intricate compartmentalization of eukaryotes, each suited to their own ways of life.
Then we toured the eukaryotic cell, the dynamic plasma membrane controlling entry and exit, the supportive cell wall in plants, the coordinated network of the endomembrane system, Golgi, lysosomes, vacuoles, handling synthesis and transport.
The powerhouses, mitochondria generating energy, the kitchens, chloroplasts capturing light, the ribosomes building proteins, the cytoskeleton providing structure and movement.
The specialized structures like cilia and flagella, the crucial role of centrioles and animal cell division, and finally the nucleus, the ultimate control center, housing the genetic blueprint organized into chromosomes.
It truly is the fundamental structural and functional unit of life. Everything happens at the cellular level.
Absolutely.
Yeah.
And if we connect this to the bigger picture, the elegance and efficiency packed into these microscopic units are just astounding. Realizing that everything an organism does, thinking, moving, fighting disease, ultimately traces back to molecular interactions within and between these cells is, well, it's profound.
It really makes you think.
It does. And perhaps a final provocative thought for you, the listener, to ponder. As our understanding of this intricate cellular machinery gets ever deeper, especially at the molecular level, how might this knowledge continue to reshape our future? Think about medicine, disease treatment, maybe even bio-inspired engineering. Where could manipulating or understanding these fundamental units take us?
A lot to consider there. The potential seems enormous, maybe even a little daunting.
It's certainly a space to watch. Keep exploring, keep asking questions. The world inside the cell is endlessly fascinating. There's always more to learn.